them by comparing them, for instance, with museum selections, collections, but it is a very a job that takes a long time. And also there you can use a technique which we call meta-barcoding, where you just isolate the DNA from your collection of all trapped species, and you sequence this mixture of DNA. You know it's a mixture, but you sequence everything, and then you try to find back the individual species. And I will talk about barcoding a little bit more later, but on the ecosystem level, this is a technique that comes in handy. So in case people were still wondering why you need all these millions and millions of DNA reads, you can also use that method, that enormous quantity of data you can get from these next generation sequencing techniques, for sequencing samples of which you already know that it's going to be a mixture of species, like a soil sample or water sample, or all these trapped arthropods you found in your trap. So then all these DNA reads, they represent a mixture of species. Now another example would be, and I think this is really the case when studying large carnivores, they're often quite difficult to spot and even more difficult to take a sample. You can apply very opportunistical way of sampling by, for instance, collecting fecal samples. And it's often not that easy to identify the species in the field. Sometimes you have some nice footprints, like on the bottom picture, which may help. But often it's quite hard, and you can just collect fecal samples or maybe hair samples, and you use the DNA in that. And I put a small part of a publication down on the screen as well, and it's a study where they used scat samples to show the range of leopard and cheetah in southern Algeria. So even though the actual animal is not spotted, you can still, by collecting these kind of samples, you can show their presence, but you can also get some insight into their range. Now, if you collect scat samples, you have some information about let's say, the owner of the scat sample, but also what's inside of the sample. And there are more and more studies using next generation sequencing for diet studies, and this is a very nice example where they used the next generation sequencing for studying the diet of a leopard cat in two different parks in Pakistan. And these two circle diagrams, they show the diet composition in these two different parks. And I don't want to go into the details, but you can see from all the colors that are used in the diagrams, the diet composition is quite different. And in the past they would do that by studying scat and isolating the hairs from the scat sample and comparing the hairs with the library, for instance. But the drawback of that is that you often only identify one species, often the most recent prey or the biggest prey. And using next generation sequencing in diet studies can actually give you a much more, on a finer scale, much better picture. So these are some examples on how to answer ecosystem level kind of questions with using molecular techniques. Now, if we're going to go more to the species level, one thing that is used quite a lot is barcoding. And barcoding is just a method where they use a short genetic marker to identify the species. And you have large databases on the internet, like GenBank or like BOLD, where they have all the sequence information of the different species in there. And you have not only the sequence information in there, you often also have information of a voucher specimen. So if a species was described from a specific individual that is somewhere in a natural history collection, you can get all this information out there in these databases. And they're just publicly accessible. And it's also the way you would use them in, for instance, in a diet study, if you would have a whole lot of DNA reads coming back from analyzing the diet from a SCED sample, you would just use all your DNA reads and compare them with the information which is in these databases, and based on that you can identify the different species. Now, if you're not only interested in identifying species, but you're also interested in the relationship between these different species or these different genera or different families, you're doing more phylogenetics. And we've seen this before already a little bit, but this is an example of a tree showing the genetic relationship, the phylogenetic relationship of all the cat species. So this is another way of looking at DNA on a species level. And the third application would be if you would be interested in, for instance, cryptic species. And this paper shows a jackal. At least everybody always thought it's a jackal, but when they looked at it on the DNA level, they suddenly found that you can identify two groups. And even though they look pretty similar to the untrained eye, or to my eye at least, you can make a distinction genetically. So this is a way in which they found a cryptic species, something of which they always thought it was a very normal common jackal, suddenly was something else. So if we're gonna zoom in even further and we're gonna look within a species, this is the example I used before, our own lion study, and I just put the example of the phylogenetic tree up there. So this is a phylogenetic tree not comparing the relationship between species, it's a phylogenetic tree comparing the relationship between different populations. And what we see is we find a quite clear distinction between the northern part of Africa and the southern part of Africa. So that's the reason why I put these two maps there, because all African lions are considered to be a single subspecies. So taxonomy says that they're all more or less the same. But if you look at it on a genetic level, we actually find two groups. Now, one of the possible results could be that we have to revise taxonomy. We actually have two subspecies of African lion. But before we get to that point, you could also say, well, we don't really want to bother and call it a different subspecies, but we can say, based on the genetic information, it's an evolutionary significant unit. Because it's so different, it reflects having a different evolutionary history. And based on that, we can say it's a unit that needs a separate management strategy, and we can no longer manage all African subspecies, the entire subspecies, all African lions, as one single group. We have to make a distinction there. Now, this line example was from a continental scale, but if you look on a population scale, you could go even further. You could look at the genetic structure within populations. Now, here's an example where they did that for a leopard in India, and they used a variety of genetic markers to kind of reconstruct the gene flow, the connectivity between different protected areas. And they made an overview, and it's illustrated by the arrows between the circles. The larger an arrow is, the more gene flow there is, the more migrants you have going from one park to the other. And what they did is they compared contemporary gene flow with what they estimated would be the historical gene flow. And this can give you very nice insight in what important corridor areas are, how well a corridor area is functioning, and also where the difficult things are when you try to conserve a species in a certain landscape. And if it's possible to identify individuals even on the individual level, it's also possible to look at kinship. And this can give you some information about your population structure. And in this paper became quite famous as the paper of the cheating cheetahs, where they studied the litters of African cheetah, and they found that 43% of all litters were actually half brothers and half sisters from each other. So apparently the female cheetah would mate with different males, and the offspring would have different fathers. So this is quite interesting. This is something they didn't really observe before, but using genetic data made this suddenly very clear what the strategy of the cheetah would be. And what's very important for conservation-related issues is the problem of inbreeding, and we talked about it before. This is something which is important to pick up quite early in the process, because then it's still possible to rescue a population or to intervene and manage it in a way that you can avoid further inbreeding. And in the first lecture I already mentioned that inbreeding is a very well-known problem in the Florida panther, and this is the picture of a Florida panther, and they did something really interesting there. It's also a little bit of an ethical question, but what they did was they found that the Florida panther was very strongly affected by inbreeding because the population had gone down very quickly as a result of habitat fragmentation and degradation. And they actually thought that although there were still some individuals around, that there was no way of actually rescuing this species. So what they said, the only thing we can do is we can only rescue the species or the subspecies by getting up the genetic level to a decent level, by hybridizing it with a different subspecies. Of course, it means that you kind of lose the integrity of your Florida panther, your genetic integrity of your Florida panther. So you kind of lose a subspecies, but you rescue at least a part of it. So I want to take a little bit a closer look at that example, but first to go quickly back to the theory. So we already said in small and fragmented population we are dealing with different things. We're dealing with inbreeding and we're dealing with genetic drift and gene flow. Now in the case of the Florida panther, there was only one population left, so there was no possibility for gene flow to another region. And all these things combined, they led to the loss of genetic diversity and also to the accumulation of deleterious mutations. So what they saw was that the going down of the genetic diversity actually really resulted in a lower fitness, and these Florida panthers had all kinds of strange deformations and other things that clearly were not a sign of a genetically healthy population. So as we said before, that genetic erosion, this loss of genetic diversity on the large scale, it leads to the loss of adaptability. But on the short scale, it leads to birth rates going down and death rates going up. And that's exactly what we've seen also in the extinction vortex. And once a species is in there, it's quite difficult to get it out. One way to get it out, and that's what they did in the Florida panther, is by saying we have to hybridize, as I said, with different subspecies. So in 1995, they brought in eight female panthers from Texas, being a different subspecies, and all the red dots indicate these introduced individuals. And you see the other dots are all pink and yellow, which are the original Florida panthers. But if you look 20 years later, or 12 years later, you see that there are a lot of orange dots, and that's the offspring resulting from this hybridization between Texas panthers and Florida panthers. So not only did the population go up threefold, but they also measured that the heterozygosity went up and survival and fitness measured improved. And another way of showing it, and it's actually in kind of a similar shape as with the prairie chicken, is in these graphs. So on the x-axis is the time, and on the y-axis you can see the yellow and the pink Florida panthers that are not doing too well. Heterozygosity is going down, and at the point of introduction of the Texas panther, the heterozygosity goes back up. So this is a quite successful example how they kind of restore the population, but it was quite controversial because it led to the hybridization of two subspecies. Main thing is that a lot of species, they are pretty good at avoiding inbreeding. There are all kinds of strategies to do that. The only thing is you have to give these species a chance, and in small and fragmented populations, in a fragmented habitat, often they don't have the chance anymore. So this is just a selection of things that can be used as a strategy. So one paper is about dispersal in wild bird populations to avoid inbreeding, and up there again is the cheetahs that mate with different males to keep the diversity in the population high. And there's also a paper about wolves where they tracked different wolves, and they found out that wolves with high levels of heterozygosity actually had a better mating success than wolves with low heterozygosity, or even better than that you would expect by chance. So a lot of species, they have their strategies of coping with it. Now, to go back to the lion example, somebody who tried to make a model to define what it is that actually avoids inbreeding in a lion, and they made a model and tried to include all kinds of parameters, and what they found is that a population needs to be minimum of 50 prides, which is rather big, and they need unrestricted male dispersal, which means that you still need the connectivity to other populations. Even though you have 50 prides, you still need the connectivity. And then he looked at all the natural parks, all the protected areas in Africa, and he found that the thin vertical line shows what is a sufficient area according to the model to avoid inbreeding in the case of the lion. And you see that most of the parks, because it's a frequency distribution, you see that most of the parks have an area that is much lower than that. So it means that if we don't think about management, on the long term we really have a bad chance, and inbreeding will occur in all these populations. And I think that's the case with a lot of species, and especially with large carnivores, because they need a lot of space. Now, if you take a little bit a closer look, in southern Africa there are many game reserves. Some of them are private owned, and they all have been restocked with wild animals. And 37 game reserves, they contain over 500 lions, so we're talking about big numbers here. So this is not a zoo, this is a more or less natural area, but it's often a fenced natural area, and it's managed quite intensively. So in theory, it kind of behaves as a zoo if you look at it from a population genetics perspective. Now, if we take the example of the Madikwe Game Reserve in South Africa, it's actually the fourth largest lion population in South Africa, and the second largest reintroduced lion population. The largest introduced lion population was actually introduced from Madikwe, but it has grown much bigger over the past few years. And what they did in this game reserve is they introduced lions, and they tried different ways of introduction. That's what it says in the table. So once they tried 2 females and 1 male, and once they tried 2 males. So they tried different types of introduction to see how these individuals would establish themselves in the park. And after that they followed them. And what they saw is that over the course of the years, density went up, which kind of makes sense because lions are quite happy to breed as long as they have enough food. So if you provide them with food, they will reproduce quite easily. So density went up, but the inbreeding coefficient went up as well, and it went up very rapidly. So that's kind of interesting, because it means that even if you restock a park, that within within a few years you can run into serious trouble if you don't do anything. So another thing I find very interesting is, apart from the population level and looking at it at the point of inbreeding and genetic diversity within the population, I find it very interesting to see where these different lions they used for reintroduction came from. And when we go back to the table, we see that most of the lions came from Pilanesberg, which is quite close to this area, which is also in South Africa. But we also see that 2 of the lions came from Etosha, and Etosha is located in Namibia. And actually, this is something we really see in our data as well, because you see in Southern Africa, there's also a small area with the grey stripes. And this is also an area where we find a combination of haplotypes, and as we don't really see that there is a connection to one of the other groups of lions we describe, we really think that this combination of haplotypes is the result of people, maybe for private collections even, getting individuals from another country, from another region, and releasing them back into the wild. And we know that that happened quite a lot, because at a certain point a lot of people had private collections, and then they decided, because of the connectivity, we should take down all the fences and we should make it one connected population. And that's what they did, which in principle is good. But if you used individuals from a different country or from a different genetic group, that is of course something we're going to find in our analysis, because it's a quite unexpected pattern. So the different levels, again, to look at these things. Now, we already talked about captive populations, and I want to give some examples really from the zoo. And in the case of the lions, it's a bit messy. For instance, in tiger, we have defined different subspecies, and the zoos kind of follow that. But in the case of the lion, there is no stud book for African lion, so there is mostly no information about which zoo is exchanging animals with which other zoo. And one thing was kind of interesting, because what was considered to be a separate subspecies of lion is the so-called Barbary lion, which went extinct somewhere in the 20th century. And there are some zoos that claim that they still have Barbary lions in their collection. And that, of course, is very interesting, because we know it's not there anymore in the wild. And from some of these zoos, there is some pedigree information, but if you look a little bit closer, you see that there are always some missing data, and you can never be too sure about what is in the population now. So a few years ago, somebody did a pretty nice research where they collected samples from different museums. And there are different museums that have lion skulls or lion skins from Northern Africa, really from the natural range of this so-called Barbary lion. And they isolated the DNA from it, and they compared it to the zoo lions that were supposed to be Barbary. And what they found is that there's actually no match. So all these lions that are in the zoo and are called Barbary lions, actually no Barbary lions at all if you compare them with the real thing. And this is something that happens quite often in zoos. And I did something similar for the tiger. So you may recognize that we have a bar plot from the structure analysis I talked about before, and we also have this network. And what they did is they sampled a lot of tigers in the wild, and they also sampled a lot of tigers from zoo populations. And they tried to get a complete picture of the diversity which is in there. And what they found is that some zoo populations are pretty pure. They represent a pure subspecies of tiger, for instance. But there are also quite a lot of zoo populations that show a lot of admixture, which you can see in the bar plots, where one bar has many different colors. So even though the zoos sometimes claim that they manage their populations following pure genetic lines, this is not always the case. However, what was interesting is that when you look at the network, they found some haplotypes, they found certain sequences that they didn't find in the wild. And assuming that they did a pretty thorough job sampling the wild tigers, it means that in the wild already some genetic information was lost and it's still there in zoo populations. And this is mainly the case for the Amur tiger. So this is quite interesting to consider, that even in maybe the natural populations has lost something, there's still maybe a second chance by looking at the zoo populations. Now to continue with the tiger, one of the very interesting things you can do also with these genetic methods is wildlife forensics, and I think this is also really an upcoming field. And in this paper they describe certain SNPs, certain point mutations that can be used to identify not only the tiger as a species, but also different subspecies. So if you find somewhere, or customs find somewhere, material like a pelt or maybe bones or something, and you're not sure what it is, this is a method, genetic methods exist to try to identify not only what species it is, but also what subspecies. And they've done this quite a lot. So the first thing would be to see what species it is, and this is something that happens quite a lot at Leiden University as well. And this is just a selection of some recent publications where they try to monitor or get an insight of the collection and trade of wild orchids in Nepal. They also looked at certain Indian snake root and Hoodia. So they try to identify certain species, maybe because they're protected and over-harvested, or maybe also because they're poisonous in products. So it's a type of barcoding, but it's a type of barcoding you use with a forensic goal. Now, if you have a good reference collection, then you could even try to find back the source population, and this is what they did here. This is based on a microsatellite study where they sampled African elephants throughout the continent, and whenever there was an illegal shipment of ivory found, I think this is the Hong Kong ivory, where they found a huge pile of tusks, and they tried to find back the source population. And here in this case they found the source population to be Gabon, where they just what they did is they analyzed the microsatellites from the illegal ivory and they compared it to all the data they had from the wild populations. Now, this is something we are also starting up now at Leiden University. We have a good connections with customs, and every once in a while customs finds some traditional medicine, some pills or some powders of which they expect that there is lion or tiger in there. So this is really something we are setting up now, and we will try to get these samples to our lab and use next generation sequencing to not only identify the species which is used in the product, but also to find where this originally came from. And we have a SNP line now in Leiden. This collection of machines I quickly showed in the previous presentation, and it has a huge capacity for gathering data points. So I think even if there are many products available, we can get a good insight where it originally came from. So I hope that this also in this way can contribute. So to more or less summarize these things, how can we use genetics or molecular techniques to contribute to conservation-related questions? One thing is to resolve taxonomic uncertainties, like not only the cryptic species, but also finding different genetic groups within a group which is considered a single subspecies, measuring genetic diversity, on a species level, but also on a population level, in which you can also detect the risk of inbreeding, for which you need to make a management decision to avoid that. Defining management units, and also guide successful breeding in captivity, as we've shown. And successful breeding in captivity, I think, that really has also potential to contribute to conservation of the species in the field. So this was an overview of how you can use the different genetic tools we talked about before lunch, how we can use them in conservation. And if there are any questions, I'd be happy to answer them.
The role of maybe outbreeding depression in all this translocation that happens, like especially with let's say the Texas and the Florida panther. I guess given that they were mainly very climatically very similar, there was no potential outbreeding depression. But do they check for that before they begin this, translocation process?
Well, first I think in a species like a panther with such a big range and much potential for gene flow, unless humans come in and fragment the habitat, outbreeding depression is probably not very likely to be a big problem. In this particular case, it was the only way to rescue this Florida population. So I think that's the reason why they went through with it. But it was a very controversial decision. Yeah, but I would personally, I would always say, we sometimes get requests from African governments with questions how to restock their own lion populations. I would always be very much in favor of using individuals from neighboring populations and not a population somewhere across the continent.
When you are taking the scats as a sample, and then I believe you will have all the fragmented DNA, not the complete genome, and there are millions of bacteria that come from gut, and there are millions of other stuff that are present in the environment, and you are collecting that scat and trying to figure out what the complete genome would look like. So you will get lots of pieces of DNA, and how would you make out that it belongs to the organism and it does not belong to the organism?
Well, what we do is for the scat samples, they often contain very little DNA and is very strongly fragmented. So one of the things we did therefore, we were really focusing on mitochondrial DNA because it's present in more copies. Now it's by using the primers we use to select the region of interest, we always, apparently they were pretty specific primers because we always amplified lion or another cat or carnivore species. So we never had really to deal with bacterial contamination there. We had to deal with bacterial contamination when we did Illumina sequencing. We used no scat samples for that, but we used blood samples and tissue samples, and even there we found a lot of bacterial reads. But if you use a reference sequence, you kind of filter out those reads because the bacterial reads do not match the reference well. So you end up with the matched reads, you continue for your SNP analysis, and all the non-matched reads are basically thrown away, and they contain all the bacterial reads and other things you don't want to have in there.
You said that eDNA tries to figure out what organisms are present in the environment. So
Yeah.
for that, the reference sample would not work, and there are several unknown organisms.
Yeah.
And how would you, there's no primers for them, nobody has discovered them, but you still try to figure out what is present. So I was confused about that.
Well, for instance, if you are interested in the animals present in an aquatic system and you isolate the DNA from water, you would basically apply a method like barcoding. You would select a barcode marker, a specific gene or a specific region in the DNA, and then from all the DNA reads you have from that all amplified barcode markers, you could compare that to a database. And I think in general it's always good to kind of focus your research question. It's very hard to sequence everything that is in there. And I guess the same thing is with microbial communities, that you would also focus on a small piece that is kind of representative for microbes. And as long as as long as you have suitable references in your database, it's pretty easy to find it out. If you would detect a new species, then you would just find that you don't have a suitable reference in your database.
Where we go for the paternity test and some kind of criminal cases to solve this kind of criminal cases, we normally use the mini satellites. Especially in this forensic wildlife, what kind of markers you prefer? The mini satellites, microsatellite, or SNPs? One question is that. Second one is there any, could you please elaborate the genetic rescue, the process of the genetic rescue?
Okay, so the first question was what genetic markers to use in wildlife forensics. I mean, it depends on your research question. So barcode markers are very suitable if you just want to detect which species is in this commercial product or this product that was found. If you want to trace a source population, they have done it very effectively with microsatellites. I think the best reason to use SNPs is that for microsatellites you still need a decent quality and decent quantity of DNA. And often, depending on how the wildlife product, how it was processed, the DNA can be very fragmented in there as well. And the good thing about SNPs is that the SNP is just a single position. So as long as you have this single position in the flanking regions, which can be very short pieces of DNA, you can still use the SNP marker there. And at this point, it's not used that often yet, but I think SNP markers can become the main marker for using for doing wildlife forensics. Yes, I do, but you need to you need, of course, to have the SNP information there. So, for instance, we developed it for lion, and I'm pretty sure it will work for tiger and maybe some other big cats. But these are all things that are in progress, and we need to test it. But if you're working for different species for which there are no SNP data present yet, it's a quite laborious and expensive thing to set it up. On the other hand, that's the same case for microsatellites, but microsatellites have been developed for more species yet. But I definitely think that SNP is the future in this, because once they are developed, it's also quite cost effectively that you can screen many samples. The best thing is to keep in mind this extinction vortex, and this extinction vortex, you see that genetic erosion, loss of genetic diversity leads to these demographic factors, and the population becomes even smaller, and the genetic factors play even a larger role in the smaller population, etc., etc. And it's very hard to get out of this extinction vortex. So the best thing you can do is make sure that this genetic diversity goes up. You cannot really affect the demographic factors that much because low fecundity or high mortality as a result of genetic erosion is quite hard to counteract. But what you can do is you can manage a population in such a way and mimic gene flow by introducing other individuals to get that genetic diversity back up, and by that it kind of creates an escape from this extinction vortex.